Next step is the fat of ammonia. Means, as we discussed earlier, the plant absorbs the ammonia as well as NO3 minus. Now this ammonia at the physiological pH in the plant, ammonia get protonated and it is converted into ammonium ion. This ammonium ion is highly toxic, so it do not accumulated in the plant. It immediately assimilated and it formed the amino acid. How the amino acid get synthesized? So there are two steps, reductive amination and the second one is the transamination. By these two processes, the ammonium ion is converted into amino acid. The first, in reductive amination, it requires the α-ketoglutaric acid. This α-ketoglutaric acid, with the help of ammonium ion and NADPH, plus it requires the enzyme glutamate dehydrogenase, it converts glutamate, H2O and NADP. This glutamate is the first synthesized amino acid. So first step is reductive amination, in which alpha-ketoglutaric acid and ammonium ion in presence of NADP and glutamate dehydrogenase enzyme convert into glutamate. Now this glutamate, it now acts as a amino donor. It is now act as a amino donor, and the next step is the transamination. This glutamate ion act as an amino donor which donate the amino group to other keto acid. This is the keto acid, it's an amino acceptor, and this is the amino donor. So this amino donor donate this ammonium ion to the keto acid, and the keto acid is converted into amino acid, and the amino donor convert into keto acid. This process is continuous and it will synthesize the different 17 amino acid. Clear? So transamination means the amino donor donate the amino group to the keto acid and this keto acid is converted into amino acid. Now in plant there are two most important amides are present, named as asparagine and glutamine. Asparagine and the glutamine, these are the two amides. From where these amides get synthesized? So these amides get synthesized from the aspartic acid and glutamic acid respectively. Okay, asparagine is synthesized from the aspartic acid, glutamine is synthesized from the glutamic acid. Actually, the aspartic acid, the structure of aspartic acid is this. It has the hydroxylic group and one amino group. This hydroxylic group is replaced with the another amino group. This hydroxylic group is replaced with another amino group, and this aspartic acid is converted into asparagine. Similarly, the glutamic acid, this is the structure of glutamic acid, has the hydroxylic group. This hydroxylic group is replaced with the amino group and it forms the glutamine. So, this asparagine and glutamine both have the two amino group. This is the one amino group and this is the second amino group. Here it is the one amino group and here it is the second amino group. So, in the amide has more nitrogen atom than the amino acid. Amide has more nitrogen atom than the normal amino acid, and this amide get transported via xylem from one part of the plant to another part of the plant. In some legume plant, the nodules, in soybean like plant, the nitrogen export in a form of ureates. Clear? So this is all about the fat of ammonia. Okay, let's discuss again. The fat of ammonia means the plant absorbs the nitrogen from the soil in a form of ammonia. This ammonia, as the physiological pH, it is converted into ammonium ion. This ammonium ion is highly toxic to the plant, so plant never accumulate the ammonium ion. Immediately this ammonium ion is converted into amino acid. How this amino acid is formed? There are two processes. One is reductive amination and the transamination. In reductive amination, alpha-ketoglutaric acid with the help of ammonium ion and NADPH, it is converted into glutamate with the help of enzyme glutamate dehydrogenase. Now this glutamate, it is the amino donor. It donates the amino group to the other keto acid. And this keto acid is formed the another amino acid. This step is called the transamination. In plant, there are two main important amides are there, and that is asparagine and glutamine. This asparagine and glutamine, they are synthesized from the aspartic acid and the glutamic acid. This aspartic acid, this is the structure of aspartic acid. In aspartic acid has the hydroxylic group replaced by the amino group, and in glutamic acid, hydroxylic group is replaced by the amino. So always amide has the more amino atom than the more nitrogen atom than the amino acid, because here aspartic acid has only one nitrogen atom and asparagine has the two nitrogen atom. Glutamine has only one ammonia. Here it is 2, 1 and 2. So amide has more nitrogen atom than the amino acid, and this amide transported from one part of the plant to another part of the plant with the help of the xylem. Some legume plant like soybean, they export the nitrogen in a form of the ureates also. Okay, so this is all about the nitrogen metabolism and the nitrogen cycle. Clear? Okay, thank you.